Thank you everyone for coming. My name is Ivan Liachko. I'm one of the founders and chief scientist at Phase Genomics based in Seattle, Washington in the US. So today what I'm going to tell you about is a method that we've developed that enables the culture-free discovery and host attribution of viruses, plasmids, antibiotic resistance genes in metagenomes. So, at Phase, we're all about genomes. We do a lot of genome assembly. And of course, in order to get to a genome, you have to start with sequence data. And that sequence data comes in the form of some sort of short or relatively, slightly longer reads, which then have to be assembled into some form of contigs, which then you try to turn into chromosomes. And this is difficult enough when you're dealing with a single organism like a plant or a human, but it's even harder when you're dealing with metagenomics, right? So the challenge with metagenomics is that you now are dealing with a complex community where you have many genomes all together. You have sequences that are coming from different organisms, and you don't necessarily know where they're coming from. And so it's very difficult to know which sequences belong to which organism. It's very difficult to track mobile elements like plasmids and phages because they're often not part of a genome. If you have multi-chromosomal genomes, it's very difficult to know which chromosomes belong to which organism. And so as a result, without having bugs in databases, it's very difficult to reconstruct them de novo. Of course, the way that this is traditionally addressed is using metagenomic binning, right? Which is a very powerful suite of tools that have been worked on for a long time that basically uses information such as coabundance of reads, tetranucleotide frequency, k-mers, other things to try to take these complex soup-like mixtures of reads and contigs and separate them out into their respective genomes. The problem is that as powerful as this is, and it's really quite amazing how much you can do with metagenomic binning, it still has limitations, right? So this method, these methods are prone to contamination because again, you're assembling genomes, you're binning them based on inference, based on how, coabundance, based on k-mers, but you don't really know that sequences belong together or not. You can do scrambling experiments where you can take, this is just a synthetic community of known organisms that you can rescramble. 10% of the genome gets scrambled, 20% of the genome gets randomly scrambled. And then if you run CheckM, is a software that looks for completeness contamination of genomes, it still calls most of them high quality, even though we know that they're all jumbled up. So it's prone to contamination. It picks up, host, like if you're doing a human microbiome sample, you will sometimes pick up host sequences. It doesn't work on mobile elements like phages and plasmids because they don't follow the same rules as genomes do. So they don't follow the same abundance rules and they have different sequence characteristics. It often doesn't work on eukaryotes and strains of the same organism or similar species because they might be, if they're present in similar abundances, their sequences are very similar together. And so, what would be nice is to have a method that lets you bin metagenomes using some sort of direct information of which sequences belong to the same organism. It would be great if it worked on prokaryotes and eukaryotes at the same time. It would be great if it could connect mobile elements with their hosts without culturing and without single cell sequencing, without having to sort individual cells. It would be nice if you didn't have to buy a special machine just for this. And if it could get around very delicate sample treatments like trying to extract high molecular weight DNA, you can't freeze cells, these sorts of things. And so the method I'll tell you about today fits a lot of these categories. And what it is it's based on another tool, a tool that was originally developed to look at the three-dimensional folding of genomes. So there's this suite of tools called proximity ligation or chromosome conformation capture. The most common of these is called Hi-C. The way it works is you take cells when they're still intact and you treat them with a cross-linker, it's formaldehyde. So you take intact cells, treat them with formaldehyde. What this does is this creates physical junctions inside the cells which will capture DNA molecules that are next to each other in physical three-dimensional space. You can then fragment this cross-linked chromatin, exposing these like little ends, and then you can use a method called proximity ligation to ligate these sequences together. This is a little bit like when you're trying to clone a plasmid, and the plasmid will want to close in on itself, right? And so in this case, you get this chimeric junction between this red molecule and this blue molecule. And then later you can isolate them through a series of steps that I'm not showing here. You can isolate these red-blue chimeric junctions, and when you sequence them by Illumina, you know that whatever this red sequence on the left, whatever this blue sequence on the right, they were physically next to each other in three-dimensional space. And because you're doing this millions and millions of times with Illumina, you're essentially counting how often every locus is touching every other locus. And so you can generate these giant matrices of interactome data, and you can extrapolate that data into 3D models of genome. And that was the original intent of the technology. But you get like really useful information that the authors, I think, didn't originally intend. And there are two things that proximity ligation gives you. It tells you how far and how close together every pair of sequences is on the genome, right? It's basically a genetic map, because the further two sequences are apart from each other, two molecules are apart from each other, the less they're going to touch in three-dimensional space. And so you can calculate it backwards. You can think of it backwards. If you're closer on the chromosome, you're going to touch more. And so if you know how much something is touching, you can figure out what's far and what's close. And we use that to scaffold plant animal genomes. But more relevant for this talk, this technology tells you which sequences begin their life inside the same cell within the mixed community. So, and I'll go more deeply into this later. So the bulk of what we have done in the last years, if you look at our publications, is we use this technology to assemble genomes. And so you basically, if you're trying to scaffold a genome for a plant, for an animal, these are humongous genomes. And so you need something like this to basically replace genetic mapping. So it's a way of scaffolding full chromosomes and haplotype phasing them. And this method is something that's been very well established. I won't talk about it. I'm happy to talk about it offsite. But it's sort of, here's sort of a gratuitous slide of a bunch of the like Nature papers and things that we've published. But as I mentioned, the real value that this technology brings to the metagenomic community is that it can be summarized in the following statement. Anytime two sequences interact by this method, by Hi-C or 3C, you know that they must have started out inside the same cell, because the crosslinks that joined them together happened on intact cells, right? We create these crosslinks before the cells are broken. So they're happening inside of cells. So if two things are cross-linked together, they must have been inside the same cells at the beginning of an experiment. And so, in other words, if you have a metagenomic community like this one, and you sequence it, what you're going to get is you get this alphabet soup of shotgun sequences. And but if you have proximity ligation data, it will create these chemical, it'll create these junctions inside the cells, and these junctions will be between DNA on the same chromosome and between different chromosomes, and it will enable you to figure out which sequences belong together. Right? If these sequences are touching each other, they belong part of the same organism. And important note is that it works on both intra and interchromosomally. So it'll work not just on the chromosomes itself, it'll work on multi-chromosomal genomes, it'll work on plasmids and hosts. And so this is the first ever proof of concept experiment we did. We published this back in 2014. The idea is we took a synthetic mix of 12 yeasts, we shotgun sequenced them, and we did Hi-C on it. And we took the assembly and we binned it with Hi-C data. And what you're seeing is these all the dots in this figure are contigs, and the edges, the lines connecting them, represent Hi-C signal. So if we didn't do proximity ligation on the sample, if it was just shotgun, this would be a gray field of dots. You can see that Hi-C is able to take them and separate them into these nice clusters. And of course, if we look at them, these are these clusters represent the organisms that we put in. And we saw that even in those very early embryonic days of this technology, it was very accurate. These are all eukaryotes. They have, many of them have plasmids. They have 10, 16 chromosomes apiece. And so you can really do a lot. This was very obvious that you can really do things with this technology that you can't do with normal sequencing. And you can even go further. So once you've separated these genomes apart, remember, when you have high-C proximity ligation data for a mixed community, you're essentially, you have this data now for every member of this community at the same time. So you can do all the other things that you do with high-C. So for instance, all these purple contigs belong to this one organism. Well, we can actually scaffold this organism into full chromosomes, which is what I've mentioned in previous slides, where we use it for plants and animals. You can do the same thing for fungi. We've actually done a ton of fungal genome assembly and scaffolding with this method. You can map centromeres in fungi with this data. It's really pretty neat. And so it was very promising when we started doing this, and so we turned it into an accessible commercial product. We were calling it Proximeta for proximity-guided metagenome assembly. It works like this. Just like I said, you take a metagenome sample, your microbiome sample, you cross-link it with formaldehyde, you do proximity ligation to generate these kind of junctions, and then you use that data to deconvolve metagenomes. And here's the rest of the talk. I'm going to show you examples of what sorts of things we've published and some unpublished, and what other cool things we can do with it. So first of all, our binner, Proximeta, takes both normal binning methods and high-C binning methods, puts them together, and tries to get sort of the best of both worlds from these assemblies. And as such, it basically is guaranteed to outperform any other tool. So it outperforms, on the left here, this figure is showing this is from a fecal sample, sort of how many genomes we're extracting using different tools. And so you can see Proximeta, you're getting tons of these really high quality genomes. That's one of the other things that's really neat about this, is that sometimes you can get lots of bins for a sample, but when you have these high-C data sets, it's really good at getting really high quality, really complete bins as well, especially for novel organisms that nobody's ever seen. And so, you get, no matter sort of what you do on sort of using normal binning methods, Proximeta outperforms. You get all these new genomes. That's what these orange dots are for a given sample, things that are only identified by this high-C method. This is a screenshot from our platform. So what we do is when we deconvolve a sample, we will basically, for every mag that comes out, we get a bunch of metrics for it. So the way to read this chart is, these are all the mags, clusters, genomes, bins, however you want to call them, along the x-axis. The blue dots represent completeness according to CheckM. Orange dot is contamination according to CheckM, so you want that to be low. And these gray bars are measures of novelty. So we run all sorts of taxonomic software on these things, CheckM, pPlacer, MASH. And so we try to see if this is something that we can easily map against. And so if there's something that has a gray bar, it means we can't map against RefSeq that bin. And so what's really nice about the high-C method is that it is really good at assembling genomes for things that are novel, that this is all done with no a priori information. And so you're able to get genomes that nobody's ever seen before without, kind of too much effort. It just automatically assembles them. And so this is from a data set that's based entirely on Illumina sequencing, right? There's no culturing here. There's no cell sorting. There's no long reads. This is only Illumina. But you say, well, I'm a fancy rich scientist. I have long read data. Say, great. Long read assemblies can generate really amazing outcomes. Here's an example that was just recently published with our collaborative work with folks at the USDA. This is a very deeply sequenced with PacBio hi-fi sheep poop sample. And what you will notice here is, first of all, just the sheer number of genomes that are coming out of this. So there are about 400 genomes, 400 mags from this sample that are over 90% complete. And then the other thing, and this is to some extent my favorite, is that this field of gray here basically means that almost every one of these bins, almost every one of these mags that we're pulling out does not have a good match in RefSeq. So there's an enormous amount of novelty that this method can pull out simply by answering the question, sequence A, sequence B, did they belong inside the same cell at the start? Did they touch each other? And so armed with this information, you can do some really, really cool things. And we always try to sort of push the envelope with what we can do. Of course, one nice byproduct of having these better genomes is that it enables you to do much better functional annotation of these samples. So, for example, if you take the fecal sample that I showed earlier on, and you just do metabolic annotate for metabolic modules, what we find is that so all the squares are metabolic modules. The yellow ones are ones that we find in the Hi-C data set. The black ones are ones that we find only in the DOS tools assembly, so the conventional binning and dereplicated with DOS tools. Blue is if we don't find it in either, and green if we find it in both. And so one of the things you see here is that there are a lot more yellow than black dots, meaning that you're just getting a lot more biochemical information out of these when you process them with Proximeta. We also have data that I'm not showing, where we spot check these, looking for realistic kind of false positive rates. It looks like most of the black dots, at least in this case, probably are false positive. Something like 3/4 of them, whereas the yellow dots are only about 15% false positive as far as we can tell. So what it's saying is that because the quality of the assemblies, you're getting more genomes, more metabolic modules, and they're more accurate, higher quality. And this is just on the right, this is a blow up of one particular pathway. And then, your orange boxes are the modules that you're only getting out of Hi-C assembly. There's another angle, though, in addition to just being able to get improved MAGs, improved genomes out of metagenome samples. One huge blind spot is, as I mentioned earlier, is mobile elements, is phages and plasmids. And it's because these are difficult to bin, they're difficult to assemble, and they jump around from host to host. And so it's when you extract DNA from a sample, you no longer know where these plasmids came from because the DNA is not connected to the genome. And so it would be really nice if we can connect mobile elements to their hosts. And that's one of the things that, as I mentioned before, Hi-C allows you to do. So here's an example of a super complex sewer wastewater sample that we processed. It's got lots of MAGs. They're not even assembling because there's too many of them. But in addition to looking for the genomes, we can annotate this assembly to look for antibiotic resistance genes, for plasmid sequences, and see which ones connect to which hosts, because they will touch each other, right? And that gets you data sets like this. So we published this with folks from University of Idaho a couple of years ago. What you're looking at is from this one sample, if you just take the proteobacteria, to simplify it a little bit, the host genomes, these bacterial MAGs, are here in the middle. And out here in these concentric circles are plasmids, integrons, and antibiotic resistance genes. And so when you see a tick mark like this, that means that this antibiotic resistance gene was present inside of that host, okay? And, so you see there's tons of them. There's all these clusters. If you see a line like this, that means that this antibiotic resistance gene was present in many hosts. So it's moving around. And if you get something like this, like a line in this direction, that means that whatever this bacterial host is, it had all these plasmids at the same time. So you're able to not just look at sort of make one-to-one relationships, but you can tell promiscuous mobile elements, you can tell multi-drug resistant strains, things like that. There's another paper we published similar in the rumen sample, but this one didn't just look at antibiotic resistance, it also looked for the host of phages. As I mentioned, you can detect when viruses are present inside of which host. And this was originally shown by Romain Kozul's lab in Institut Pasteur in France. And what you're seeing here is these are bacterial hosts from this was a mouse fecal sample. And these are the phages, and you can see that you can figure out the host phage relationships directly out of a fecal sample. And so what I'll talk about in the last few minutes of the presentation is a new thing that we've developed along those lines. So when you do this cross-linking, you get phages connecting to the host, right? This is how you can figure out which mobile elements are present inside of which hosts. But you get more than that. So when you cross-link these samples, you are going to get interactions inside of a phage genome, and you're also going to get interactions between the host and the phage, right? In other words, if two contigs are really coming out of the same phage, they will interact with each other physically, but they will also interact with the same host, right? And you can use those two pieces of information simultaneously to enable us to do something that's really novel, which is actually accurately binning phages. So not just assigning, like you annotate a phage and you can figure out who the host is, but actually binning a little genome for the phage. This is something that's super difficult using conventional sequencing. So we developed this method, which is, by the time you're watching this talk, it's going to be in bioRxiv. We call it the proxy phage, but essentially what it does is it takes proximity ligation data and some sort of metagenome assembly. It will do the binning and it'll annotate viruses, and then it will essentially bring those two pieces of information together. It will simultaneously look for the host contig interactions between viral sequences and bacterial mags, and it will also try to basically bin phages together and put them put those two bits of information at the same time, and it allows you to create viral mags and simultaneously map them to their hosts, where they came from. And then, so you get these amazing data sets that look like this. These are contigs, and they're in a stripe like this because, so all of these, each one of these little pie slices is a different viral mag, a viral genome. And the lines inside of them, these are contigs that belong to the same host, right? So they line up. So all of these contigs belong to the same viral mag, and they are hitting the same host. In addition, you can look not just these are the mags that we reconstructed. We can actually figure out who their hosts are, just like we did before. So here, these are the phages, and these are the bacterial mags. And so you can not just detect like individual placements, but also if you have co-infection, multiple viruses infecting the same host, you can see that like this. If you have one phage that's infecting multiple different bacterial mags, you can see that here, right? And you're getting all of this information at the same time with one data set. New mags, new viral mags, and the same thing with plasmids. Well, I'm not talking about plasmids, but the same thing is happening with plasmids, and you're connecting them at the same time. We did a lot of benchmarking to make sure this is accurate. I don't have time to go too deeply into it, but long story short, we use computational tools such as CheckV to check the accuracy and completeness of these mags, but we also use long read assemblies, right? So if you have a long read PacBio or nanopore assembly where phages are basically single contigs, we can compare our short read viral bins against these and figure out how accurate we're doing. Long story short, it seems more accurate. We were surprised by how good it is. Only about 4% of viral mags that come out of proxyphage look to have significant amounts of contamination. We're also able, like from a fecal sample, we can get 75 phages that are 95% complete using only Illumina data. That's the key here, is that we're able to get these viral genomes without long reads, with just short reads. If you have long reads, it's great, it's even better, but you don't have to have them. This is a method that works on all sorts of samples. So here's some like sheep and humans and cows and sewer, and there's soil and all sorts of other things. The method works on pretty much anything that can be subjected to metagenomic Hi-C. And so just to end my talk, I will mention briefly an experiment. We can start doing experiments now. Everything I've shown up to this point has been one sample, right? But here's a time course. This is a time course of fecal samples taken from an individual after finishing antibiotic treatment. So day 0 was the last hit of clindamycin, and then it was recovery. And so what you see is over time, you're starting to see more bacterial mags popping up. At the same time, new viral mags, new viral contigs, and new viral mags are popping up. So presumably the bacteria are bringing new phages into the population. At the same time, AMR sequences and plasmids seem to be going away. right? So, which is what you would expect from recovery. But now, of course, we can go deeper. We can actually take these samples and look at the host mobile element associations as these samples progress through time. And that's what's shown here. I don't have time to go into it. Feel free to talk to me as a human, and I will answer your questions about this technology. And just to remind you guys, all of this does not require any culturing, no a priori information. You don't need high molecular weight DNA. You don't need any special machinery. It's just a kit. We sell these kits. The software comes along for free on our analytic platform. I would like to thank our collaborators and other folks that we work with. And if you're interested, please reach out and feel free to ask me any questions. Thank you.